big impact. Oh, this is going to be interesting if we can't advance on the screen. Okay, so I'm just going to give you a quick overview. We're going to talk about forage fish today and what they are and why they are important. Specifically, we're going to look at Washington forage fish. We're going to look at how people in Washington study those fish. And then I want to talk to you about the Whatcom Marine Resource Committee data trends. Okay, so forage fish are an ecological, not a genetic group, and they're sometimes called bait fish. But either term can be broadly applied to this diverse assemblage of species that are, in many cases, only related through ecology. And this includes a greater range like animals such as krill and squid. Forage fish are generally small and planktivorous, and they are highly fecund, which means that they have lots and lots of babies. They also tend to school, and they are right in the middle of the food web. So because of this, they are that vital conduit between our primary producers and our higher level consumers. So because of this place in the food web, forage fish can be an indicator for ecosystem health. For example, forage fish abundance has been linked to the success or failure of shorebird nesting colonies, to salmon runs, and to marine mammal survival. And when forage fish abundance drops, other fish like salmon smolt may be the next item on the menu for many of these predators, especially harbor seals. So forage fish have supported commercial and recreational fisheries, and they currently account for about 37% of marine harvest by weight. And they've also been very important to First Nations and to the native tribes of North America. So these are just some pictures of recreational harvesters doing dip net fishing on the Columbia River, and then some commercial harvest here in Puget Sound. And like I said, these fish have been extremely important to the culture and to Native Americans in Washington state. So there are some challenges with monitoring these fish. They are important both economically and ecologically, but there are challenges because we don't really understand how our human activities impact their abundance. Forage fish stocks fluctuate dramatically in response to ocean conditions. In this example, let me highlight these colder years here, the Pacific Decadal Oscillation, which is the top figure. And the colder years favor Peruvian anchovy. But if you look at the bottom figure, you'll see that the warmer years tend to favor sardine. So scientists need to be able to separate the effects from human-induced effects. And recent studies have shown that fishing pressure can make these declines more dramatic or prolonged. So here in Puget Sound, there are a lot of concern over forage fish abundance because even though we have very little direct estimates of abundance, we do have a lot of indicators, and some of them are not looking so promising. So in this slide, I wanted to highlight some of the bird declines that we've had in Washington state. Birds that dive and forage for fish in the Salish Sea, including this western grebe that's pictured here on the left side, left bottom of your screen, they're 11 times more likely to experience population declines than other birds in the area. The diagram on the right shows many other species of common birds that are declining in Puget Sound, and scientists are increasingly concerned that declines in forage fish, particularly herring, may play an important role. So let me just describe what's happening in this right side diagram here. The green bubbles or circles are population estimates that were done in 1978 and 79. And then the pink dots are population estimates in 2014. And you can see that some of our birds, like the long-tailed duck, have seen a 94% decline in their populations over that time period. And the western grebe have been reduced by 99%. Austin, are you still there?
I am, yes.
Just making sure. Looks like I lost you guys, and you never.
Oh, I just stable. Oh, okay, no, I just muted and stopped my video.
Perfect. Okay, so moving on, we are seeing some very dramatic declines in bird populations, and that is very concerning to scientists and people throughout the state. So what are some of the factors that could contribute to the declines of forage fish in Washington waters? Well, these are some of our usual suspects, the first being climate change. The rise and fall of forage fish populations are often correlated with ocean conditions, like I showed earlier, and changes in the frequency and the severity and duration of some of these events may result in shifts in the distribution or abundance of our forage fish species. And over time, these species may become less abundant. We also have overfishing and bycatch. So these fish, like I said, are schooling, and schooling makes forage fish vulnerable to overfishing. They're easy to find and they're easy to catch in large numbers. And some are also caught in large numbers even when they're not being targeted. Another concern that we have is poor water quality. It's been shown to negatively impact many fish species, including forage fish, especially the early life stages. And the nearshore species that I'm going to talk about today are often exposed more frequently to contaminated water because of their proximity to human development. And degraded habitat and habitat loss. This might be a little surprising because everybody assumes that the ocean is so huge, but a lot of species, especially in Puget Sound, rely specifically on that very narrow band of beach at the edge of the water, and they use that to lay their eggs. And these species must compete with human interests for that space on the beach. So let's talk a little bit more about some of the common marine forage fish in Washington State. This is a very diverse group of species, and they have a variety of life histories, and they also occupy a variety of habitats. For example, spawning habitats for these species, the ones that I'm going to discuss in greater detail today, they can kind of be divided into three general types. We have our pelagic, like anchovy and sardine and mackerel. We also have our anadromous species, like Eulachon and longfin smelt, and they spawn in the rivers. And then we have our nearshore spawners, and these are fish like surf smelt and Sand Lance to its own specific location, and also their spawn timing. So as you're going to see, we even see differences among the same species in some of these aspects. So this is a challenge for monitoring forage fish. A challenge is finding their habitat and where they're spawning and how human activities might impact their abundance. So how do we study forage fish? Well, globally, the most common approach to monitoring these fish are fishery-dependent methods. And typically this relates to some sort of catch per unit effort. So essentially a commercial fishing vessel will go out into the ocean, and they will take a certain amount of time to catch a certain amount of fish. And we can record this time and amount of fish caught, and we can look at how much is caught for how much effort is put in. Okay, so what if you don't have a fishery, or the fishery is too small, like many cases in the Puget Sound, or they're driven by regulatory or economic forces that prevent a representative sampling? Well, if we have that, like we do in Puget Sound, then we have to turn to fishery independent monitoring. And this can take many forms. Some fishery independent methods are very similar to our fishery dependent methods, but some are exceptionally different. So, because forage fish are so diverse, there is not one fishery independent survey to monitor them all. This is the NOAA boat, the Reuben Lasker, and this boat is used to do an acoustic survey and on that group of fish that I described as the pelagic spawners. So, pelagic spawners. First, let me tell you a bit more about them. Both anchovy and sardine are pelagic or open water spawners, and both of these species may spawn multiple times within their lifetime. And their eggs tend to drift and generally hatch within 2 to 4 days after spawning. Sardines are primarily found on the outer coast, but anchovy occur in Puget Sound, and recently we have seen fairly high abundance of this species, especially in the last few years. But this species of anchovy are also found on the outer coast. Northern anchovy stocks have not been formally assessed since 1995, but federal scientists estimated in 2015 that the stock size was about 31,000 metric tons, based on the acoustic trawl surveys that they do. And they've recently presented data showing that that stock may have declined over 90% since the 1980s. So let's talk a little bit about this acoustic trawl survey. On the left side of your screen, you will see some transect lines. And essentially that is the boat running an acoustic survey from Canada, on Vancouver Island, all the way down to California. So an acoustic trawl essentially happens like this. A boat goes along the floor and it sends a signal down into the water. And that signal bounces back and it shows distinct schools of fish when they're in high densities. That signal is bounced back to the boat, and then researchers on that boat target those groups of fish that they saw in the acoustics, and they run a net through that group and they catch whatever fish that are found within the signal that they sent. So these fish are brought on board and dumped into totes, and then they're sorted by species. So by doing this, researchers can determine how many of what fish are in those schools of fish, and then they're able to also take biological samples of those populations. They can take maturity and fecundity estimates, as well as male-female proportions from the samples that they're getting from their trawl survey. So all of this ends up as abundance estimates. So these are coastal pelagic species, 3 that I've been talking about: the Pacific sardine, the Pacific mackerel, and the northern anchovy. And you can see the abundance estimates from 1932 all the way to 2019. So sardine are primarily found off the outer coast, and they kind of, in 1932, their populations were extremely high. They kind of had a resurgence in the 1990s. They didn't approach anywhere near the abundance observed in the 1930s and 40s. But they have since declined, and they have led to another closure, and the fishery was once again closed in 2015. So in addition to the acoustic trawl that happens on the Reuben Lasker, there's another type of survey that is used to look at abundance of forage fish. And this is called the Continuous Underway Fish Egg Sampler. So this is, the name is the same as it implies. This technology continually samples the water around the boat, giving real-time ichthyoplankton samples to the crew of scientists that are on board. So every 30 minutes, the chief scientist takes a sample and quickly scans it for the presence of eggs and larvae of the target species that they're looking for. And if the target species is present, then the boat continues its transect line until there is no detection of the target species. This is called an adaptive sampling plan. And this sets the offshore limit of the transect lines. So at one point when they were running this fairly recently, their transect line ended over 200 nautical miles offshore. So it's quite a distance that they were covering, considering that we started around 10 nautical miles offshore. So this is just an example of egg counts of sardines. I'm not sure how everybody's computer is, or if you're on a phone, it might be a little bit hard to see, but essentially it looks at the abundance of sardines in the eggs, in the water samples that they're collecting from around the boat. So this is very useful in determining abundance estimates of sardine off of our coast. Okay, so now let's move on to our anadromous spawners. So we have the longfin smelt and Eulachon. Longfin smelt generally return to spawn in rivers at about 2 or 3 years of age, and Eulachon return at about 3 to 5 years of age. And after spawning, most of the adults die, but not necessarily all of them. The eggs are attached to sand and gravel, and they generally take about 20 to 30 days to hatch. Runs of Eulachon have historically been important to a lot of our native peoples, and until recently, they were listed as ESA. There was an ESA listing in 2010. They used to support both commercial and recreational fisheries. And just as a little side fun fact, Eulachon are also called candlefish. And it's not because of their shape, it's because they are so fatty that you can literally light them on fire like a candle. Okay, so Eulachon smelt. Eulachon are a forage fish stock, and they're known to fluctuate very widely. But the trends of some of the stocks are declining, and some stocks appear to be at the lowest abundance that we have ever seen before. So the landings essentially began to plummet in 1994, and they were listed as an ESA fishery in 2010. So without having an abundance of fish, how do we sample them? So our studies have focused on anadromous spawners, we had to take into consideration how we were going to sample in fresh water. So WDFW has conducted studies that focus on the distribution of Eulachon spawning in the Columbia River and its tributaries and in other rivers along the Washington and Oregon coast. So because of the low numbers of returning adults, it meant that it was going to be difficult to document their presence, and we had to rely on collecting eggs and larvae to determine if the river was being used for spawning. So samples were collected using primarily this ichthyoplankton net, and we also used some artificial substrates to collect eggs. Okay, so the trends in Eulachon smelt. The fishery was closed, and forage fish stocks have declined, but we're using other types of monitoring to look at their populations. We did see large runs in 2014 and 15, and some small fisheries were opened, mostly so that we could get some biological samples from those. The runs are still well below historic levels, but we are anticipating that the 2020 run is expected to be better than 2019. So in addition to this type of monitoring, we've also recently looked into monitoring environmental DNA for Eulachon. So this is a quantitative way of assessing what is in our streams. And essentially the higher concentration of DNA that you get in your water sample, the more fish are in there. So we tested out this theory and this methodology in 2017 in the Chehalis River, and we were looking for Eulachon. So we conducted a DNA sample test in February of 2017, and then we ran our ichthyoplankton nets through in March. And essentially, the DNA came back with large returns of fish in the river system in February, and then 4 weeks later, we saw a large return of larvae. So there's high hopes that this might be a very useful tool for measuring forage fish in some of our river systems. So I realize I've been kind of talking fast, and it looks like let's have just a few minutes of a stretch break. And Austin, if you have any questions or anything you want to say during this time.
Thank you so much, Lisa. So yeah, if anyone has any questions, you can go ahead and use the raise your hand function on Zoom, which if you click on the participants icon in the middle of your bottom middle of your screen, it'll be on the lower right hand side. So I'll take some questions now if there are any. Otherwise, please stretch your legs, walk around for a little bit.
Okay.
There is one question. Hold on, I'm trying to get to you, Kristen. Hold on. You know what? I'm just going to unmute you because there you go. You're unmuted, Kristen. Go right ahead.
So I see you're recording this. Is this recording going to be available?
It will be, yes.
Looking at later?
Yes. Yes, and we're hoping that we'll get some help to have this be put on like a YouTube video. So it is being recorded. Okay,
great.
Yeah.
Thanks a lot.
No problem.
Austin.
Chris.
I was going to ask Lisa if how did the Eulachon and longfin smelt lay their eggs? Is it in the water? Is it in the mud, silt, whatever?
Yep, it's on the sediment. So they lay it just on the bottom of the river, and the eggs are quite sticky. So they stick a little bit to the rocks and the sand and the gravel on the bottom of the river.
Are there locations where they have their breeding areas?
You know, I don't know if we've identified any specific locations. I think maybe they just swim upriver as far as they can, or until they feel like that's a good place for their eggs to be laid in the bright habitat. But I'm not really sure about that.
Okay, thanks. Thanks, Austin.
And Lisa, there is a person here who would like it if you could discuss the Nooksack longfin smelt abundance, if possible, if you could touch on that.
Yeah, so I'm not exactly sure what the abundance estimates are. I just know that they are in that river system and we see them occasionally. Their numbers are not very high, but we don't have any abundance estimates at the moment.
Rachel, I will get to you. All right, go right ahead, Rachel.
Oh, I was just going to say, yeah, we work in the Nooksack River system on the long fins, and as you know, as far as I know, there's nobody doing any abundance estimates. But I was wondering, with your eDNA analyses, is that all being done in-house? What I mean, you have to have genomic, with the qPCR, you have to have genomic primers that are specific for the long fins versus the Eulachon. So is that all done at WDFW?
It's not. I believe that we have partnerships that help with the processing of those samples. And I think some of those are at UW. Phil Dion would be the person to ask about that.
Okay, thank you.
Okay, so are we ready to move on to some of our nearshore spawners?
There's one more person, AJS, who has raised their hand, and it looks like you've also unmuted yourself, so please go ahead.
Yeah, this is Alice Sigurdsson. I don't know why I'm AJS. I guess that my computer says that that's who I am.
Okay.
I had a question about what are hooligans?
That's another word for Eulachon?
Okay, I wondered. All right, thanks.
Yeah, no problem.
Do they still, are you monitoring them very much, by the way? Because they usually run right around, what is it, Veterans Day?
Yes, and we are, and we did this year.
Okay. Thanks.
That's everything. Go right ahead, Lisa.
Okay, so let's talk about our nearshore spawners. Okay, so the three common nearshore spawning forage fish that we have, or I'm going to talk about today, are herring, surf smelt, and sand lance. So there's an overlap between the spawning habitats for these species, but generally, herring spawn in the lower intertidal to subtidal zone on aquatic vegetation. Sand lance spawn in the mid to the upper intertidal zone on kind of a fine sandy substrate. And surf smelt spawn in the upper intertidal zone on substrate composed of primarily sand and gravel. So surf smelt and sand lance are intertidal spawners, meaning that their eggs are deposited on the beach above low tide, while herring are subtidal spawners. We need to use different methods to study their spawning habitat. So herring surveys have been conducted in Washington since the 1970s. Spawning has been documented in about 20 different areas, and spawning generally occurs from January through April for most stocks, but some stocks spawn into June. Genetic analysis of herring in Puget Sound indicate that there are 3 genetically distinct groups, and those are our cherry point stock, our squawks and pass stock, and then all of the other areas are generally grouped together genetically. Commercial harvest for herring in Puget Sound is primarily for bait, and it averages about 300 to 400 metric tons a year. So this is a pretty picture, and it's also a picture of a herring spawning event. And we also call this whitewater. So the herring come near shore and females deposit a sticky egg on vegetation, and they're fertilized by the milt or the sperm released by the males into the water column. And if you look closely at this picture, if you can see marine mammals feasting on these eggs and also birds taking advantage of this spawning event. So we do herring egg surveys, and herring surveys are conducted by boat, and we use a special rake that's dragged through the water to collect samples of vegetation. The density of the eggs on the vegetation and the area of the seafloor that's covered by the vegetation are used to estimate the number of eggs in the area. And then from that number of eggs, we estimate the number of adults that would have been needed to spawn that number of eggs. So we don't always necessarily know the amount of vegetation in the areas that we're spawning. So this is an example of the 2018 Killisett Harbor Acoustic Vegetation Survey that we did because we need to get an idea of how deep the band of vegetation is that the herring have to spawn on, so that we can make good abundance estimates of herring in Puget Sound. So where does this get us? Our herring stock trends. The Cherry Point stock was once the most abundant stock in Puget Sound. But since the 1970s, this stock has declined by over 70%. While the abundance of the other stocks has remained relatively stable, and that's a lot has to do with Hood Canal, but both the average size and the age of herring in Puget Sound has declined. But we do still have commercial fisheries for herring in Puget Sound. Fisheries tend to target our subadults, and they're used locally for the bait market. We have limited gear. It's limited Lampara nets and dip nets, and we tend to close areas during spawning season. So if you look at the map on the left, our areas that are closed during spawning season are in green. And then we have some year-round closed areas where you're not allowed to fish at all, and those are the areas in red. So in general, of the spawning stock biomass estimates, the annual harvest for herring is less than 5%. So acoustic trawls, I talked about those on the coast, but we also conduct acoustic trawls in the Puget Sound. We use a much smaller boat, and we do not get to as many places, but we also use this type of survey to determine what our herring populations are doing in Puget Sound. So let's look at our other near shore spawning forage fish. We have the Pacific sand lance and the surf smelt. So surf smelt and sand lance are special because they lay their eggs on beaches near the high tide, which means that their habitat is very close to our habitat. And this also puts them at risk. So risks to the nearshore forage fish habitat include, but are not limited to, both direct and indirect effects of nearshore development, which means that they can have direct habitat loss when a beach is built on. There's a loss of riparian cover. There's changes to drainage patterns, and there's changes to wave energy and sediment transport, especially if beaches are rip-wrapped or armored. And there's also a problem when there's an impoundment of beach nourishing sediment. And this happens when we armor our bluffs that normally would erode into the nearshore environment and kind of feed the beaches to give forage fish something to spawn in. So the nearshore has many issues, which is why our beach surveys are so important. So in addition to our Marine Resource Committee partners that we work with, we also have a crew of WCC members, that's the Washington Conservation Corps, that work with us to sample and process samples that we receive from the MRCs every month. So for those of you that are familiar with this type of sampling, we tend to use the methods that were set forth in 2006 in the Field Manual for Sampling Forage Fish in the intertidal regions. But essentially, this is what happens. When we arrive to a designated site, we assess the location for first accessibility to the crew, and then for habitat suitability. For habitat that is unsuitable, we, for example, if it's mud or hard pan, we don't collect any samples there, but we do collect habitat characteristics so that we know not to go back to that place that it's unsuitable. So at suitable sites, we collect sediment samples across a 100-foot section of beach, and that sediment is later sieved through progressively smaller sieves, and we keep only the sediment that is in the 5mm sieve that's on the bottom. Any eggs are then extracted from the sample, and this is where our methods differ from the 2006 protocols as we've adapted over time. It used to be through this winnowing method, and then it was sent to the so once the blue bowl vortexing method has been done, we send those samples to the lab, and those samples are processed to look at surf smelt and sand lance egg presence, egg abundance, and egg developmental stages. And the data that I'm going to show you for our MRCs, I just want to remind you that egg presence is indicated by 2 or more eggs found in a sample. So this may be very familiar. I'm hoping that this is familiar to many of you. This is the forage fish spawning survey map that I send out each month or every other month, depending on how many samples we have. And I just wanted to walk you through this. This is the one that I sent out in October and actually November of 2019. And it shows all of the samples that were taken over October and November. So during that time period, we had 226 surveys conducted. 8 of those sample events had surf smelt only. 5 samples came back with sand lance eggs in them. And none of the sites had both sand lance and surf smelt eggs in them. But you can see the light blue dots are places that were sampled that had no eggs in the samples after we processed them. Okay, so I want to narrow it down just a little bit more to an area that you guys are the most interested in, and this is our North Basin. The North Basin runs essentially a little bit south of Anacortes all the way up to the Canadian border. And this is data from sampling that was done between 2016 to 2019. The red dots are anywhere that a survey was conducted, and the yellow dots are anywhere that there were surf smelt eggs. And this is just for surf smelt because it gets way too busy with surf smelt and sand lance. So we're just going to look at surf smelt data for this presentation. Okay, so let's break it down a little more. In 2016, we all conducted 326 sampling events, and of those, 13 samples had surf smelt spawning in them. There was a peak in June and October, and there was also spawn detected in this area in April, May, and July. We did not have any samples collected in February or March, which is unfortunate. And there were no spawning events detected in January, August, September, November, or December for 2016. So the map that I'm showing on the screen extends a little bit farther down than the North Basin. But I do find it interesting. That's the Whidbey Basin down there, and the green stars indicate where spawn, surf smelt spawn was found. It's kind of interesting to see the Whidbey Basin population as well as the North Basin population as we look through these maps. Okay, so this was 2016. The red dots are surveys that happened with no spawn ever detected at those sites, and the green is places where surf smelt spawn was found. Okay, so this is our 2017 map. There was an increase in numbers of samples that we collected in this year. We collected, among all of us, 464 samples, and there was a total of 19 surf smelt spawning events detected. There was a peak in spawning in January. There was also spawn detected in February and May, and there was no spawning events in April, June, or December. 2018, it looks very, very bare in comparison to the following years, and that's because of a sampling change for WVFW. We were looking at historic areas that originally had forage fish spawn detected, but we hadn't seen any in the last 10 to 15 years. So we wanted to focus on those places and see if we were missing spawn. So we were not able to get up as far north as we would have liked in 2018, but there was a total of 100 samples collected, and there were 7 positive for surf smelt. Remember that the bottom area is not in the North Basin, so those numbers are not included in this, but do note that there is a lot of spawn that does happen in that Whidbey Basin. During 2018, spawn was found in January and August, and there was not any spawn detected February, March, April, May, September, or October, or November. But like I said, we had some limited data collection during this time. And then 2019, we did increase our sampling. Again, we were able to go up to the San Juan Islands. However, that sample event all happened within the same month. There were 149 samples collected, and the peak spawning was detected in March. Spawn was also found in June and July, but there was no spawn found in August, September, October, or November. So just a little bit better representation. This data was actually given to me this morning. So we have a group that is processing this data set from 2016 to 2019. And I literally just received this, and I thought, hey, I might as well put it into the presentation so that you guys know where and when spawn is being detected on the beaches. So because of beach data collection, we can look at some of the trends in spawn timing, which allows us to, well, possibly limit nearshore beach projects during critical times of forage fish spawning. This information also helps us understand population dynamics, and it allows us to adjust fisheries if we need to. So if you're looking at these diagrams, please note the number of samples on the left axis are all different. And the 2019 data, our March sampling event, that primarily focused in the San Juan Islands, so that, that number of samples is actually off the chart. But I wanted to bring it down so that we could see that, for example, in July, more of the samples that we collected in 2019 in July had spawned than did not. Same with August and September. So this gives us a very good indication of when the forage fish are using our beaches and when they're spawning on it, and when we need to protect this critical habitat to make sure that the eggs that are spawned on their beaches have the best chance of survival. So like I said, having this data helps us. We do have a commercial smelt fishery in the Puget Sound, and a lot of these fisheries are regulated and based on seasons. So they're open weekly from Monday through Thursday. We do restrict fishing effort between daylight hours, which is 6 a.m. to 10 p.m. normally. Also have a 60,000 pound annual limit. So if you look at the map on the right, there are areas that are open the entire year, but a lot of the areas we close down to help protect some of those stocks that are trying to spawn on our local beaches. We also have a recreational smelt fishery, and this is also open weekly, but it's modified or shortened, and it is only open Friday through Tuesday. We also restrict fishing effort between 6 a.m. and 10. And as of 2019, we have a new license requirement. So prior to that, there was no requirement for smelt fishing in Puget Sound to have a license. But recently, our regulators and legislator and managers have decided that now this is becoming important. So just a few more stats. Our current harvest levels are about 40% below our 22 average, and this is because we chose to reduce the amount that is allowed us to take because we are concerned about some of our forage fish. But most of this harvest occurs in the Whidbey Basin, and in the maps that I showed you with all those stars farther south, not in the North Basin, but farther south, this is why a lot of the fishery happens there, is there's a lot more forage fish. All right, so in addition to the beach and gravel samples that we do around Puget Sound, we also conduct beach seining and Lampara netting surveys. And these methods are great because they're relatively low tech and they're inexpensive and they're easy on the fish and the habitat. So that, and we can also sample shallow areas. They do give us biological samples, so we can look at gender specific and natural mortality and fecundity estimates. We also can conduct tagging experiments when we catch fish in this way. So we do have a tagging experiment that we are hoping to conduct this year. I don't know what the status of that currently under our COVID-19 restrictions, but we do have plans to do a tagging study for smelt, and we will see if that happens. But the importance of forage fish is becoming more clear to policymakers and managers, and it's even making it into the news. This is something that was posted from the Eyes Over Puget Sound, where they saw schools of forage fish. The Orca Task Force has also helped shine the light on our marine food web and some of the limitations for salmon survival, which impacts our orcas. So in this screenshot, or in this thing that was posted, these are actually anchovy. And anchovy have been in high abundance in Puget Sound in the past few years. And some researchers think that this might be due to our warmer water conditions. This is the beach seine that we did last year, and there are literally millions and millions of anchovy caught in this one set off the beach. So anchovy in the Salish Sea are becoming a little more interesting to us. And during our beach seining operations, we have recently noticed an uptick in the proportion of sets with anchovy in them. We compared our data to data from the Strait of Georgia, and they came to similar conclusions that we did during some of the same years. They also have some data points that we're missing because we didn't do our beach seining during those years. So this is fairly interesting, and we took this one step farther, and we looked at the Nisqually River steelhead smolt survival. So these fish are tagged, and as the fish go out of the Puget Sound, the tags are detected at various locations throughout Puget Sound as they make their way out of the Strait of Juan de Fuca. So we can see where their survival rates were higher and lower, and we also can relate this to our anchovy abundance during these years. So it may be that when there's a lot of food around, a lot of the predators don't predate upon the steelhead smolt as much as they do forage fish in the system. So this is a picture of the marine mammals feasting on anchovy. And even though forage fish are small, their health and their populations, the health of their populations directly impact the entire health of the Puget Sound. And it's something that we all need to be concerned about and make sure that we know what are happening with our forage fish populations. So with that, I guess I just want to ask if there are any questions.
Thank you so much, Lisa. I'll just kind of monitor the participant list here. I'm not seeing any hands raised or any messages in the chat. Give it another minute.
Well, I love it. I mean, I answered every.
Oh, here we go. We have a chat. Awesome presentation. Thank you. Many people are saying thank you. Fascinating. Great presentation.
It was my pleasure.
Yeah, thank you so much. Could you perhaps just post your contact information one more time so that if folks do have a question and would like to get in touch with you, they could do that via email? I think
Definitely.
it was.
Let me see here. There you go. So there is my email address, and that's probably the best way to contact me at the moment. But I also do want to say thank you to all of the volunteers and the Marine Resource Committee people and folks and helpers that provide us with our samples on a monthly basis. We really appreciate that. And this year, 2020, is going to be exceptionally challenging because our agency staff are not allowed to go out and do the surveys that we want to, so we're missing time. But I know some of our MRCs are still collecting, which is very, very helpful. And that data will become very important as we try and assess what's happening.
Yeah, thank you so much. I still do not see any hands raised. So I guess that is it. Thank you so much for your time, Lisa. This was a really fascinating presentation. I appreciate you willing to share this with the community. So,
My pleasure. Any questions that you have in the future, please email me and I will get back to you.
okay. Thanks a lot, Lisa. And thanks everyone who joined.